Most people think of protein as a dietary component coming from meat, dairy, or whey powder. However, proteins are actually amino acids organized in different ways. Living things can make most of the amino acids used to create proteins, but 9 different amino acids do come from foods such as meat, nuts, and some vegetables. Once these amino acids are arranged and folded in thousands of conformations to create proteins, they serve as the driving force for almost all of the work done in and around a cell. Cell biologists and other scientists have studied how different proteins work to understand the processes going on in living things, to understand why certain disorders and diseases happen, and to potentially find ways to cure those disorders and diseases. This presentation covers information about proteins in general and then looks at an example of a protein, called tubulin, whose function is vital to the inner workings of a cell and may lead the way to treating some types of cancer. Although there are around 20,000 known proteins, all proteins can be classified by 4 levels of protein structure: primary, secondary, tertiary, and quaternary. The order of the amino acids in the amino acid chain constitutes the primary structure of a protein. Often, the amino acid chain is thought of as the backbone or ribbon, and the way this ribbon will repeatedly fold forms a pattern either in the shape of a beta sheet or an alpha helix. This repeating pattern is called the secondary structure. In this figure, an amino acid backbone can be seen repeating and forming an alpha helix. Some parts of the protein fold irregularly. Irregular folds break the repeating pattern of secondary structure to form loops and turns. All of the irregular folds throughout the protein make up the tertiary structure. And there are some proteins that can be categorized by quaternary structure too. When smaller proteins come together to form larger proteins, the smaller proteins are called subunits. Quaternary structure is the way these subunits are arranged. All of these different proteins with unique three-dimensional shapes have just as unique functions. 4 categories of proteins can be found just in the cell membrane, including enzymes, transporter proteins, receptor proteins, and anchor proteins. Enzymes are proteins that catalyze cell reactions. This means they are proteins that allow reactions to proceed at comparatively low levels of energy, and many enzyme proteins only catalyze one reaction. There are a lot of reactions going on in the cell, and so a lot of different enzymes are needed. In parts of the cell that are not the cell membrane, there are proteins that function as storage, transport, and antibodies. Others still play a huge role in structure and support of cytoplasm. The cytoplasm has a complex network called the cytoskeleton, which is composed of microtubules. Microtubules are made of proteins. The protein that polymerizes into the filaments that form microtubules is called tubulin. Microtubules are able to shift and change formation, allowing for cell division, chromosome segregation, and vesicle transportation. Microtubules and tubulin are found in all eukaryotic cells. In Figure 2, an image of a section of microtubule is shown as what looks like a complex connection of different parts that form a hollow cylinder or tube. Tubulin allows for the potential formation shifting of microtubules because tubulin is flexible. The most mobile regions of tubulin are found at loops, or what make up the tertiary structure, while tubulin is less flexible around the binding sites. After 30 years of research, a team of scientists completed a model for the structure of tubulin. The researchers discovered a compact molecule that has 3 functional domains that are binding sites for nucleotides, certain drugs, and other proteins. One of the problems with picturing tubulin was the way tubulin forms into filaments. And in order to solve this problem and for tubulin to be pictured as a sheet, zinc was present during polymerization, preventing the formation of long chains and filaments. This flat formation allowed for imaging by electron crystallography. Tubulin was found to have two polypeptide chains that differ in sequence but are almost identical in structure. Both polypeptide chains, called monomers, have two beta sheets surrounded by alpha helices. The monomers share almost half backbone identity, including 405 amino acids that are the same. The secondary structure is made up of 33% alpha helix and 21% beta sheet. Loops and turns are the flexible part of tubulin, and so tertiary structure changes. Alpha tubulin and beta tubulin are each subunits that make up quaternary structure. These pairs of alpha and beta are called dimers. In this figure, there are two clumps of squiggles and loops that can be seen, and each are one of the dimers. Dimers come together in groups of 13 to form the microtubules. The interest for the structure of tubulin was sparked when tubulin was discovered to bind to a drug called Taxol. Taxol will keep a cell from going through mitosis and has been effective in treating some types of cancer. When tubulin is binding with Taxol, the tubulin is no longer a flexible protein because Taxol causes stabilization. This figure shows the tubulin binding with Taxol at a beta subunit. Taxol is the green part of this picture. If tubulin structure was better understood, it was thought that a more precise version of Taxol could be created to bind only with tubulin in cancer cells. Taxol is still being researched, as well as how the drug can be effectively and efficiently extracted from its natural state. A scientist, Eva Nogales, working on figuring out the structure of tubulin, also said, "The knowledge gained from this model should be of invaluable help in understanding the microtubule system in the cell." When tubulin is distorted, it has been linked to disorders in both plants and animals. The Institute of Plant Sciences at the University of Bern found a correlation between tubulin and semidwarfism, and weak stems of teff crop. Tubulin has also been found to affect dog diseases that influence platelet formation. A congenital defect in humans called circumferential skin creases Kuhn's type is also correlated with the tubulin defect. Proteins are invaluable for life, function, and structure on a cellular level. As scientists understand more about how proteins work and what protein is responsible for what process, things like curing cancer seem more plausible. Tubulin and Taxol are just one example of the importance of the work done by cell biologists. Looking at all the work that proteins are involved in might even make all those dark green veggies go down easier. All sources from the presentation, including the figures, are listed here.